Hi there, and welcome to session two on disease and the immune system, essentially, how we deal with disease. Let me just check. Can you hear me okay? Last session, we started looking at all the different types of diseases because they can be grouped into different groups. So we had a look at things like hereditary, genetic, idiopathic. We're having a look now at infectious diseases. This is a really big topic because there's so many different types of pathogens, and we've looked at different pathogens in different units. We had a quick look at last session at some viral diseases. So we talked about flu, influenza. That's our main concern in terms of viruses, just because of how catastrophic it is if we have a really bad flu season, how many millions of people succumb to it because it's a respiratory disease. So respiratory diseases are by far the worst in terms of us humans because of the way they spread so easily and quickly from person to person. We have moved on now to have a look at some bacterial diseases. So the one we stopped on was cholera. So I'll just recap where we got up to because we were going to have a look at how cholera is treated. So cholera is an infectious disease. It gives very watery diarrhea, which can very quickly lead to dehydration and death in vulnerable people. So old people, children, babies, really badly affected by it. It's transmitted by eating food or drinking water that is contaminated with a bacterium that causes cholera, and it's mostly in water. It tends to be in places where there's poor sanitation, usually during crowding or war or famine. So after natural disasters, and there's sort of tent cities that are housing displaced people because of some natural disaster like war, or that's not natural, but some disaster like war or famine or flooding as well, more frequently with climate change. Essentially, you need clean water. That's all you need to do. So that's easily solved. It's just a matter of getting the infrastructure in place. So there is no reason why we shouldn't be able to do that now, like today. It shouldn't be happening because we know what causes it. We know what the risk factors are. We know when it's likely to occur. So we should be able to prevent it happening. That's not always the case. Unfortunately, there's always politics involved, and look at what's happened in Gaza, for example, being unable to get things like medication and supplies into these places. So it is really difficult. So signs and symptoms are basically the signs and symptoms of dehydration: rapid heart rate, loss of skin elasticity. If you pinch the skin, it stays in a sort of pinched position for longer than it should, and you get dry mucous membranes, your eyes feel dry, your mouth feels dry, your nose and inside your nose, everything feels dry. You feel thirsty, you get muscle cramps. It can cause death in babies very quickly. So it is a very serious condition. So once you've become infected with cholera, there's two things you need to do: treatment with antibiotics to get rid of the infection, and oral rehydration, not dehydration, that should say rehydration, oral rehydration therapy to make sure you take waters on board. So you can give people water, but you also need to give them a little bit of either salt or sugar to help keep that water on board, or otherwise it will just pass through and get excreted from the kidneys. That's what oral rehydration therapy is. It ensures that the body will hold on to, so it's a bit of sugar in the water usually, or a little bit of salt. It sounds counterintuitive to give salt, but it makes you hold on to the water then when you drink it. It has to be carefully regulated because you can't have too much salt, or otherwise you get more dehydrated, but you need a little bit of salts to help you to hold on to that. Cholera is one of the main causes of severe dehydration. In 2010, 3.5 million people contracted an infection by cholera. That's a lot. And obviously, the death due to dehydration is a risk in all of them. So, other bacterial diseases that we should know about include gonorrhea. That's a sexually transmitted infection, or STI. It can grow and multiply very easily in warm, moist areas such as the reproductive tract. Or it will also grow in the mouth, the throat, the eyes, and the anus. So any mucous membrane it comes into contact with, any non-skin type tissue. The reason gonorrhea is of interest at the moment, particularly is there are antibiotic resistant strains of it around. So it's really important. We can't treat it with antibiotics. So it's really important that the population is aware of it. So it is difficult to know that you have gonorrhea sometimes. Some men have no symptoms at all. However, some symptoms that men might see is a burning sensation when urinating or discharge from the penis, which can be white, yellow, or green, or swollen testicles. So it's really important if you get any symptoms, you go to the doctors, because it's not to say you'd have an antibiotic resistant strain, it could be treatable. In women, symptoms are often mild and some have no symptoms as well. You can get non-specific symptoms, which might just feel like a normal urine infection, or you can get burning sensation when urinating and increased discharge. So not very pleasant, but something that needs to be treated, and of particular importance because there are antibiotic resistant strains around at the moment. It's now recommended that you have 2 drugs for the treatment of gonorrhea. The CDC, that is the Centre for Disease Control, now recommends, that's the American World Health Organization, NHS would look after it in the UK. World Health Organization is obviously a global body, but in America, the CDC now recommends dual therapy, 2 drugs, because of the rise of drug-resistant strains. So because it's transmitted primarily through sexual contact, it can be prevented using barrier methods. So condoms during sex is a way to prevent that. A couple of questions then. What's the role of the cell wall in bacteria? You'll have to think back to remember that one. And which of the following has the function of being the site for most of the cell's reactions? Again, you're going to have to think back to that. And how is gonorrhea spread? So have a look at those and see if you can remember the answers. The top 2, you might have to be thinking back a little bit because it was a session ago that we were looking at that. And then I'll pop them up on the screen. If you're watching the recording, just press pause and then you can press play when you're ready to see the answer. Well done, guys. Some good answers coming in there. It's nice to see. The cell wall in bacteria helps to maintain the cell shape. It's the cell membrane that controls what enters and exits cells. The cytoplasm is the site for most cells' reactions. Actually, it's quite often the little organelles within the cell's cytoplasm, like the mitochondria, but essentially it's not the nucleus. It's within the body of the cell. And gonorrhea is spread through sexual contact. It is cholera that is spread through contaminated water sources there. So another bacterium that we're asked to know about on some of the units on this course is tuberculosis. Mycobacterium tuberculosis is the full name. When you're writing out names of Latin names of species, so like Homo sapiens, which is humans, or Mycobacterium tuberculosis, which is this bacterium which causes TB, you write it in italics. And then the first time you write it, you write out the whole name. So Mycobacterium tuberculosis. And then if I was to talk about it again, I would just write M. tuberculosis. So you don't have to write the first bit of the name out every time. And it's always in italics. So Mycobacterium tuberculosis is the bacterium which causes TB. It's a very serious infectious disease and it primarily affects the lung, but it affects the bones as well. It can get into the brain, it can get into the kidneys, it can get into the bones. It's very common, like the spine. So an important thing about this is that it requires oxygen to grow and survive. It's quite hard to grow on a plate in the lab. So it's slow growing, so 2 to 6 weeks to form visible colonies on culture media. So that means it's difficult to diagnose, or it was historically difficult to diagnose, because you get a diagnosis by growing things in the lab. It's got a waxy cell wall, which means it's difficult to treat because it's highly resistant to desiccation or drying out, disinfectants and antibiotics. So it's difficult to treat and it's difficult to kill, essentially. Humans are the main reservoir for this. It is also found in cattle, in deer, in badgers. So cattle are a bit of a problem. They spread it around the wildlife as well. Transmission via coughing from person to person. Pulmonary TB is in the lungs. So anything in the lungs is a problem. It has some ability to survive inside macrophages, which is similar to HIV. It means it can survive some of the immune cells that are trying to clear it. So that's a problem. It can cause not a particularly bad initial infection. So just maybe you think you've got flu or a cold and you don't realize that you've actually got TB, and the bacteria can remain dormant. So you can have latent TB and can re-emerge later. Active TB, you get very sick. Chronic cough, weight loss, fever, night sweats, coughing up blood. It kills people. Particularly, you might not die from it in the UK. We've got a good, robust healthcare system, but in some countries without such good healthcare, it would be extremely serious indeed. And it's also the first diagnostic sign of AIDS. So if you've been infected with a HIV virus, it's often when TB develops, because it's an opportunistic infection. Because you've got HIV, your immune system is compromised, you get infected with this bacterium, and then your immune system has become overwhelmed at that point. So that's a good example of a bacterial infection. So we're still on infections, and here's athlete's foot, which is a typical fungal infection example. It's not very pleasant, sore, itchy, lives between the toes. It's transmitted with spores of the fungus or broken strands of the body of the fungus, if you like. You pick this up if you are in wet changing rooms, really, swimming pools. You should probably wear your flip-flops if you're around any poolside. It can be reduced by good foot hygiene, so keeping your feet clean, keeping them dry, and just wearing your sliders or flip-flops, whatever, when you're around the pool. It's very easily treatable with antifungal drugs. So this is malaria that we're going to have a look at. That's a mosquito. That's what spreads malaria. Malaria itself is caused by something called protozoan. Protozoa are tiny single-celled organisms, and they live usually in water or wet places, and they're really small. You can only see them under a microscope. Most of them just live in water or in soils. They're free living, but some of them live in the bodies of animals, and this is the one we're looking at, lives in mosquitoes and humans. It's responsible for malaria. So it's caused by a genus called Plasmodium, and it's mosquitoes that you can see there that spread the disease. So it's very widespread in tropical and subtropical regions, and it's moving further north due to climate change as well. So we're not in a malarial region, but Europe, parts of Southern Europe maybe already, but it will certainly spread northwards due to climate change. But you do need rainfall, warm, wet, moist conditions. You can prevent infection by preventing bites. That's one of the main ways to stop yourself contracting the disease. So what happens is you get bitten by a mosquito, it drinks your blood and leaves some of the parasites in your blood as part of that process. So it gives a nasty fever, you get shivering, joint pain, vomiting, anemia caused by hemolysis, so your blood cells bursting. It's pretty nasty. It's cyclical, so you'll get sudden coldness, then fever, and then it lasts 4 to 6 hours. Infection with malaria can lead to coma and death, particularly in young children. It's really nasty in little kids. So as I was saying, you get bitten by a mosquito, that sucks your blood, the parasite enters the bloodstream, that will then migrate to the liver. In the liver, the protozoa cells multiply and they change form, and they rupture the liver cells and escape back into the bloodstream. Blood cells are then infected and the parasite continues to multiply. And this results in blood cells bursting and releasing the parasites. And then another mosquito will come along, one that hasn't got parasites, maybe hasn't got the malarial bug, and it will pick up these parasites then whilst feeding to get your blood, and then it will carry them to the next person, the next host, and then they'll get infected, and this whole life cycle continues. So you need the mosquitoes to spread it, and you need the mosquitoes to catch it. So if you can avoid getting bitten, you'll avoid it completely. It can be treated. There are drugs available, but obviously prevention better than cure. Last little disease causing, it's not even an organism now, it's a molecule, prions. So what that is a load of carcasses from Creutzfeldt-Jakob's disease, which is basically commonly known as mad cow disease. We had an outbreak in the early 2000s in the UK, and it ended up with an awful lot of livestock having to be slaughtered and burned as a result. It was pretty catastrophic for the farmers and obviously the animals involved. CJD, or Creutzfeldt-Jakob's disease, is spread to people from cows with BSE, mad cow disease. It can have a very long incubation period over 10 years, and it's also possible to be transmitted person to person through blood transfusion, but we've only seen a few cases, but it is possible. There's been no recorded deaths from Creutzfeldt-Jakob's disease in the UK since 2014. We're extremely careful about the cuts of meat that we eat. In the EU, there's very strict laws about food. You're not allowed to eat parts of the nervous system and things like that. There's no effective treatment currently available for this disease caused by these prions, which are essentially proteins. So it's just essential that you don't eat meat that's been infected. That's why the whole herds of cows were taken out of the food chain when we had an outbreak in the UK. And as I said, very strict rules on which part of the animal you're allowed to eat as well. Quick look at some questions then. How is cholera transmitted? What type of pathogen is athlete's foot? And how can you treat athlete's foot? So, as usual, if you're watching the recording, press play, then press pause when you're ready to see the answer. And if you're in the live session, you can pop the answers up on the screen if you want, or just write them down if you don't fancy doing that. It does help you to just recall. Okay, good work. That was quick, guys. Well done. There you go. So cholera, contaminated water usually, and food, but it's usually water. Athlete's foot is fungal, so it can be treated with antifungals. Now's a good chance if you've got any questions. I'm just going to pop the next load of slides up for you. Where are we then? So we've had a look at some of it says, explain the ways in which infectious diseases can be transmitted and discuss specific examples. Evaluate factors which increase the risk of contracting infectious diseases and how this risk can be reduced. So one of the main routes of transmission of infectious diseases is animal transmission. We just had a look at this example with malaria. Protozoa are spread from person to person. It's not just malaria, actually. There's so many diseases that are spread by insects: yellow fever, dengue fever, Zika virus, loads and loads of diseases. But malaria is the main one. It's the one that kills the most people, and it's very prevalent. So mosquito is not the disease. Mosquito is what we call the disease vector. It's what spreads the disease. So very common insect-borne diseases in these sort of tropical and subtropical. I'm just going to show you this little video of this life cycle. Let me just make sure you've got sound. It's not going to play, I don't think, actually. I'm not sure why. What I'll try and do is I'll see if I can get you the link to that. That was an Open University video. So these are a load of slides. I used to teach a unit at Cardiff University on infectious disease. These are a load of slides from a colleague of mine who was traveling around looking at this. This is Anopheles mosquito, which spreads malaria. And he was having a look at the way people in these malarial regions try and prevent infection. So one of the things that you can do to stop being bitten, because it's very common to get bitten at night time, is you can protect your house. You can wear long clothes. So as you can see here, this guy is in a malarial region. He's got long shirt on and he's got long trousers on. But if you have a look, he's got this wire mesh around in his house. So that should stop any insects. So if you open the windows, you can still get the air in and it should stop any insects getting in the house. People in these regions often sleep underneath a mosquito net, and these are impregnated with insecticides essentially, so that the insects won't survive if they get on them. And obviously it's a physical barrier as well. You can get things called mosquito coils, which are just little, essentially insecticides, which are spread in the scent around the room. And then we've got this plant here. It's a wild plant of this chemical. You don't need to know the name of it, which is very similar to the one which is in the mosquito net. There's all ways of preventing the mosquito from hanging around in an area. And now you can get plug-in versions as well. Another thing that is done to prevent insects from reproducing in an area is fogging with insecticide. As you can see here, there's an awful lot of insecticide being added around the home and in the bushes, in the trees. Obviously, there's issues with this because it's going to kill non-target organisms. So if there's any bees or spiders, or not that spiders are insects, but non-problem creatures could be affected by it. So it's not good. It's very non-specific, so it has environmental effects. And also it tends to be not very good for people to be breathing in these insecticides either. There are things you can do to control animal transmission. You can take out the vector, but none of the methods are great. It is the problem. We were talking earlier about, in the last session, about influenza flu. This is a real problem because the main reservoir for flu influenza is birds, and they can carry the virus around really easily because obviously they can fly. So it's really easy for these bird vectors to infect domestic poultry. And as soon as it's in farm animals, it will spread to other animals on the farm, horses, pigs, all sorts of marine mammals can pick up flu from seabirds. So they can all act as intermediate hosts. So they might not be the original vector, that's bird species, but they can also become infected. So because birds will migrate, as in around the world, this is obviously a global problem as well. They can take it miles. If there's an outbreak, they can just up and fly hundreds of miles and spread the disease really quickly. The virus is shed in the feces and other bodily fluids, and obviously water birds will defecate in ponds and lakes and contaminate watery environments, and then the mammals in the area become infected really easily as well. We can control the domestic vector of flu, and if it gets into pigs, other farm animals, we can control it by basically just killing the farm animals again. But obviously it's horrible for the animals, and it has huge financial cost as well. Also, domestic pets like dogs and cats can catch it as well. So there are multiple reservoirs of flu once it gets out of the natural reservoir birds, and it can be extremely difficult to control. So this particular virus and several others actually can jump between animal species, and you end up with new strains then that can cause pandemics, which is a concern. So perfect example of this was the H1N1 flu virus. It was an avian flu. It spread really rapidly from person to person by respiratory droplets. It led to an extremely lethal pandemic, which killed more people than the war, because it happened at the end of the First World War when all the soldiers were returning home from war. So they basically took the flu with them all around the world. The second wave of this pandemic was pretty bad because people started getting secondary bacterial pneumonia, which was unusually severe and very aggressive as well. So, as we said, mass movement of soldiers, and obviously soldiers were living in unsanitary conditions because they were coming home from a war. They'd been on the front, so they were living in awful conditions. They were cramped together to return home, and they just spread the flu amongst them and around everyone they came into contact with when they got home. We're not really sure where it came from, this particular flu. It appeared in the US in early 1918. Other theories point to China. Some people say it was found in Europe. It was called the Spanish flu, but it probably didn't originate there. It's just there were troops returning from Europe. China is quite a common place for diseases to break out because and Asia, other parts of Asia as well. There are what we call wet markets there, where people buy animals still alive and they're slaughtered either in the market or you take them home to slaughter them. In Europe, that doesn't happen. We've got quite strict rules on basically processing of meat. So animals are slaughtered in an abattoir and then they're cut up by butchers. So we've got quite strict rules. So we're really quite tightly controlled on how we're killing these animals. But because there's wet markets in some Asian countries, because of this practice, they're keeping live animals close together in cages, basically, until they're bought to be eaten. So then the disease can spread really easily and really quickly, because don't forget, it's respiratory droplets, it's feces. So it's essentially they're bringing the farm to the market and in contact with the customer. So although it's one way of making sure your food is fresh, because you know it's only just been slaughtered, it also isn't great for disease. It can cause a huge problem. With the Spanish flu, genetic sequencing of victims has shown that it was an avian strain, H1N1 strain. Remember, those are the proteins on the surface, the hemagglutinin and neuraminidase. That's where the H and the N come from. And it's the proteins on the surface that change, and we can identify these flu types from the different surfaces. So the original flu came from a wild waterfowl influenza A virus. Before it caused the pandemic in humans, it has to get some adaptations, such as the ability of one of its proteins, the hemagglutinin, the H, has to be able to bind with specific receptors in the human respiratory tract cells. So there needs to be a bit of a mutation, a bit of a jump in the genetic makeup of it before it can do this. Pigs are a real issue because they can become infected with both avian flus, such as new strains that arise from this vector, this original vector of flu, and human flu viruses. So viruses which have already established in the human population, they can spread from person to person. They've got all the genetic material to be able to spread easily from person to person. If they mix in a pig with a flu virus, so a pig is on a farm, it's in contact with people that happen to have flu, it catches the flu virus. It also then comes in contact with a bird that's wild, has a wild flu, steals some of its food, leaving droplets around the pig pen. You can see how that pig could quite easily have picked up a human flu and a bird flu. They can then mix in the pig and we end up with a new type of flu. It's quite easy for new viruses to emerge because of this zoonotic nature, so spreading between animals and humans. This is what we think happened in Spanish flu. So the first wave of the Spanish flu, 1918, was relatively mild. There was a little bit of a mutation or genetic reassortment, and then there was a highly virulent wave that swept the globe and killed millions. So quite often we get flu outbreaks and everyone's, yeah, flu, it's not that bad, might be in bed for a week or two. But every now and again we get a mutation and it's virulent and it's deadly and there's millions dead. So we need to not be complacent about flu, and scientists aren't. The World Health Organization is very on this. So they're watching and they're monitoring. With this Spanish flu outbreak, there was an unusually high mortality rate in young adults, so 20 to 40. So you think usually we're saying vulnerable people, the young, the old, are the ones that end up dead. But with outbreaks, but with this particular flu, it was people that you would consider to be in their prime, ages 20 to 40. What we think was happening was they had a lack of prior immunity just by chance in this age group, because older people had some immunity from earlier strains before this age group were alive. And then they had a really severe immune response because they were young and healthy. Their immune system response was so strong that the cytokine storm to this novel virus overwhelmed their respiratory systems, and that a lot of them had severe lung issues and couldn't breathe because of the immune response was so strong. I'll try and get the links for you, but basically there's a little video here showing how we're looking for the next pandemic, keeping a very close watch on these flocks basically around the world, constant monitoring of disease outbreaks, checking the strains, and very close eye on flu. So we'll have a look if I can get these videos sorted out for you. Now, one of the major problems, one of the major animal vectors that we really have to watch are bats, because they're extremely good vectors of disease. They have a unique immune system because they can fly. They're mammals, so they're pretty closely related to us to start with because they're mammals. So that's an issue. Their cells are pretty similar. We're closely related. That's why once pigs get them, we're in trouble. We're closely related. We're also closely related to bats because they're also mammals, but they're mammals that can fly. So they have a slightly unique immune system. It allows them to tolerate viruses really well. So viruses that would just kill off loads of other animals, the bats just fly around with them. They don't even notice they're ill. If domestic animals, or I don't know, wild animals like a seal caught a particular virus, it would just be really ill and probably die. Bats just carry on living their normal life, going around and stealing animal feed off farms. They have a really robust immune system and DNA repair pathways. It's probably an evolutionary response to the fact that they fly, which is a stressful state. So they have a high metabolic rate. Flying is extremely expensive in terms of metabolic demands, and it can damage DNA. So bats have a really efficient DNA repair to cope with this. And that means they can also cope with getting really sick with a high viral load, which makes them horrendous in terms of an animal vector. Their immune systems have completely evolved to coexist with these viruses. They can eliminate them or just live with them, and they're really good carriers, so they can just fly around spreading to domestic animals. That's nearly always how it gets into humans, via domestic animals. So they will go along and feed from leftover food in the pig pen or in the chicken coop, whatever they can get, and they can get most places because they can fly. So there is a big long list of viruses that are problem viruses that bats act as a vector for. So rabies is a pretty obvious one. Ebola virus, the SARS-CoV virus, SARS-CoV-1, SARS-CoV-2. So that will include coronavirus, COVID that we just had, the MERS virus, which was pretty similar, Nipah virus, which is nasty, causes encephalitis in humans, swelling in the brain, and some Hendra viruses, which cause severe respiratory neurological diseases in humans and horses. So there is a load of problems with bats. So you need to be extremely careful if you're around bats or they're around your animals to watch out for spread. So let's have a look at rabies as an example. So rabies, it's a virus pathogen. We're talking about virus. So the reservoirs can be all sorts of animals: rats, dogs, cats, bats, monkeys. And the vectors are all of those reservoirs. What you need to do if you want to control this disease is you can vaccinate against rabies, so that's good, but you really need to control feral animal populations. So feral animals like dogs that can spread it, monkeys that you could catch it off, bats. So you've got to avoid contact with these animals. But if you look at this diagram over here, you can see there's one animal in particular which is extremely likely to come into contact with man, and that's dogs. And if you've got feral dogs which are living in the streets scavenging food, they will come into much closer contact to humans than these other animals. So the dog is very likely to pick up disease from any of these other animals that it comes into contact with. and then spread it to humans, particularly in the case of rabies, because it causes the animals to bite as well. And what animal are humans most likely to get bitten by? Is dogs. And then with all of those other bat vector diseases, it tends to go via the farm animal route. So a little look at a bat that picks up rabies and spreads rabies. It spreads it to domestic animals, cows, domestic dogs, and like feral dogs living in the street dogs. Also monkeys pick it up. Again, these pictures from my colleague who was abroad studying the spread of these diseases. So that's all the viruses and their vectors, not all of them, but a load of them. Now we're going to have a look at some of the advantages and disadvantages of the use of antibiotics. Antibiotics are chemicals that slow down or either stop the growth of bacteria. They don't affect viruses. This is a Petri dish, and what you can see there is that sort of light colour is what we call a lawn of colonies. It's lots of tiny little dots of bacterial colonies. They're so close together that it looks like a complete lawn. And so that's thousands of tiny dots of bacterial colonies. Those little discs in yellow have been soaked in antibiotics, and you can see there's zones around those discs where there's no bacterial growth. And we call these areas or zones of inhibition, or inhibition of growth. What is happening on these discs is the presence of the specific discs where you can see a zone of inhibition. The bacterial growth has been inhibited because of the presence of the antibiotic on those discs. So if we wanted to test the sensitivity of bacteria to a particular antibiotic, we could soak all the discs in a different antibiotic and then have a look at which one had the largest zone of inhibition. That would be most effective. So you can compare the size of the zones of inhibition. So as you can see, some of the discs don't work on this bacteria. So that bacterium is resistant to this antibiotic where there's no zones of inhibition. But it's sensitive to these where there are inhibition zones around the disc. So this one obviously works the most effectively because it's got the largest zone of inhibition. Because the greater the zone of inhibition, the greater the effect of the antibacterial compound or antibiotic. So you can test antibiotics this way. You can test antibacterial stuff like cleaning fluids and stuff as well, because that also has an antibiotic effect. But obviously you can't drink it because it would damage your cells as well. Now, the problem with antibiotics is bacteria evolve resistance to them. It's a problem for us because we use antibiotics to stop or slow the growth of pathogenic bacteria, so bacteria that causes illness. And when we get sick and we treat it with an antibiotic, we're actually creating a selection pressure, which means we're weeding out the bacteria that are going to die from that antibiotic, but allowing a very few that might survive that antibiotic and then produce more offspring that will survive that antibiotic. So only the bacteria with the characteristics best suited, so the ones that just about survive that antibiotic, will survive and reproduce. And then they will reproduce in the presence of that antibiotic and get more strong. And what happens then is you're producing more and more bacteria that are resistant to that particular antibiotic. Now, one way you can make this worse is by not completing a course of antibiotics when you have bacterial infection. So you start to feel better and you've been given 7 days of antibiotics, and after 3 days you start to feel fine, so you stop taking them. What's going to happen is you're going to allow some of these bacteria to survive, and you might end up with an antibiotic resistant strain then. Why this is a problem is, well, it is a problem, obviously, because we don't want antibiotic resistant bacteria, but how it becomes a problem is the bacteria can really easily spread these small sections of DNA called plasmids. Remember, there's small circular bits of DNA. They can really easily spread these between bacteria of the same species, but also just random bacteria of totally different species. They can pass on these plasmids containing antibiotic resistance genes. So it is a real problem. They don't need to reproduce, they can just pass on these plasmids. So if you get antibiotic resistance bacteria of a certain strain in a hospital, it could spread to other strains of bacteria as well. So really quick spread, not just of, I don't know, gonorrhea. We know there's antibiotic resistant strains of gonorrhea around. It's not like, oh, that's fine. You're only going to catch that if you have unprotected sex with someone. That's not the case. Any bacteria that is antibiotic resistant can pass that on to antibiotic resistance onto any other bacteria it comes into contact with. So it is a huge problem and needs to be very closely monitored because we're going to run out of treatment options eventually. So once a bacterium or bacteria, type of bacteria, become resistant to one type of antibiotic, you can just switch and use another type of antibiotic, but eventually you run out of antibiotics. We've only got certain amounts. We haven't got an infinite amount. Hopefully we'll be able to use AI to start designing synthetic antibiotics that will give us a bit more of an edge. But anytime we design a new antibiotic or develop or find a new antibiotic, the bacteria will mutate to overcome that antibiotic. When bacteria become resistant to all the common antibiotics, we try and treat them with methicillin. It's used as a last resort. And once they're resistant to all antibiotics, we call them superbugs. There are some examples like C. diff is talked about, Clostridium difficile, which is really common hospital-acquired infection. We were talking about nosocomial infections, hospital-acquired infections last session. And also MRSA, methicillin-resistant Staphylococcus aureus, is another hospital-acquired infection. There are ways of treating these antibiotic-resistant bacteria with maggots. I did some work on this 20 years ago. So what we can do is, because there's just no antibiotic with Staphylococcus aureus, there's just methicillin resistant. Once you've got past that methicillin, the last layer of defense, there is just nothing you can do. There's no more antibiotics that you can give. There is one called vancomycin, but it makes most people extremely sick. So you end up with people with Staphylococcus aureus infections that can't be treated. When you've got this Staphylococcus aureus, your flesh starts to rot. It is really serious. You can lose limbs. What you can do is use these maggots, basically what they used to do back in medieval times. And what they do is they will eat the necrotic tissue, so the dead tissue, but they don't eat any living tissue. And when they eat the necrotic tissue, that's where the bacteria is, because the bacteria is destroying the flesh. And the maggots will eat away the bacteria as well as the dead necrotic tissue. So it is possible to stay alive, but you will end up losing actual flesh, like muscle, skin. It's pretty gruesome. But we don't actually put the maggots directly into the wound anymore like they used to in the old days. What we do is seal them inside a little mesh bag and then put the bag on the wounds. But the maggots are very much alive and moving around. So it is a little bit disgusting, but it's better than losing your limbs to this MRSA, Staphylococcus aureus infection. Let's have a look at some questions. So, can we remember how bacteria reproduce? So we had a look at that last session. So it's going back a bit now because it's been a while since we had a session. Where do we find most of the bacterial DNA in a bacterium? Which of the following is not an example of a superbug? And what does being resistant to an antibiotic mean? So have a go. Think what is the most appropriate answer. And if you're watching the video, just press pause and play when you're ready to move along. Well done, some good answers there. Good work. Well done. I think that's all of the answers attempted, or all of the questions attempted between you there. So I will pop them up. So, how do bacteria reproduce? They reproduce by binary fission. So well done if you got that, and somebody got that. So well done. That's just division into two, and it's not mitosis, meiosis. That's not how bacteria divide. That's how our cells divide. So number two is a bit of a tricky one. Most bacterial DNA is found in the circular DNA. There's no nucleus in bacteria. All of this stuff is just loose in the cytoplasm. Bacteria don't have these organelles, these organized little compartments. So they don't have a nucleus. They just have that big bit of circular DNA, and it's just all scrumpled up. It doesn't look like a circular piece of DNA. It looks like on the diagrams, it looks like Mr. Messy off the Mr. Men. It just looks like all scrumpled up. The plasmids are these extra little pieces of DNA that usually contain antibiotic resistant genes that can spread from bacterium to bacterium. So no nucleus, and it's just a big circle of DNA. And then lots of little extra circles containing antibiotic resistance. So C. diff or Clostridium difficile and MRSA are, or methicillin resistant Staph aureus, are examples of superbugs. That one there, Lactobacillus bulgaricus, is actually not even a pathogen. That's quite a useful bacteria for us because or useful bacterium, I should say. We use that to make yogurt and cheese and stuff. It's the lacto bit. Think of lactose intolerant. Lactose is the sugar in milk. So Lactobacillus bulgaricus is used in yogurt production. So that's a useful bacterium. So what does it mean to become resistant to an antibiotic? If a bacterium is resistant to an antibiotic, it is able to grow. So it multiplies by this type of binary fission and survives in the presence of that antibiotic. People don't become resistant to antibiotics. We are resistant to antibiotics, or we die when we took them. We wouldn't take them if they killed us. So it's not A, which some people confuse. A few more questions then. What's the role of the cell wall in bacteria? I think we asked that earlier, but let's see if we've remembered it now. Two we did earlier as well. And number three we didn't do. That's the main one. So see if you can have a look at number three. What is the area with no bacterial cell growth surrounding an antibiotic-soaked disc called? Don't worry too much about 1, 2, and 4 because we did do them earlier. So as I was telling you last session, there's a huge amount of overlap on this unit with the microbiology unit. So hopefully that's making it a little less painful. I'll pop the answers up for you then. So the cell wall helps to maintain the cell shape in bacteria. Cytoplasm is where most of the cell's reactions take place, and it's a zone of inhibition that you get around a bacterial cell where there's no growth when it's been dipped in antibiotic. And gonorrhea we were looking at earlier, it's a sexually transmitted infection. Those we've done. Let's have a look at these. So, how is malaria transmitted? What type of pathogen is typhoid? Which type of insect can transmit malaria? and how can malaria be treated? Well done. I can see some answers coming through. Good work. I'll pop those answers up for you. Really good. 100% there as well. So it's transmitted. Correct answer, there is a mosquito vector, but it is a protozoan. You weren't that far wrong if you put C, so I said that's okay. Typhoid, bacterial, malaria is transmitted primarily by mosquitoes, and it can be treated with quinine. There are other flies as well. There's a fly called the tsetse fly, which causes a lot of problems. So it's not just mosquitoes that we need to watch out for. There are other flies which cause problems. Perfect timing. We're done. So I will see you on Thursday for the last session in this topic when we'll be going over the immune system again. But we have looked at it before, so hopefully it won't be too difficult for you. If you've got any questions, you can let me know. I'll see if I can get those video links for you as well. Okay, bye everyone.